Hi everyone, welcome to CAPSA. Today we will learn about animal cell culture. So today's topic is animal cell culture. Now, what is the definition of animal cell culture? What is animal cell culture? It is an in vitro maintenance and proliferation of animal cells that continuously grow outside of the living organisms under supplied nutrients and specific conditions. Okay? So, it is an in vitro maintenance and proliferation of animal cells that continuously grow outside of the living organisms under supplied nutrients and specific conditions. So this is the definition. Now, who developed the technique like animal cell culture technique? So, developed by Alexis Carrel in early 20th century. So this technique is developed by Alexis Carrel. Now, which type of media are used? Actually, two types of media are used. First one is liquid media and second one is solid media. Liquid media is DEM, EM or dull glucose modified eagle medium. And the second medium is RPMI1640. Solid media which is used, that is popular agarose. Okay. Now, what is the composition of media? First composition is energy sources. So, for energy sources, we use glucose and fructose. Second one is nitrogen sources. For nitrogen sources, basically amino acids are used. Third one is vitamins. Obviously, water-soluble vitamins, that is vitamin B and vitamin C. Fourth one is inorganic source like sodium, potassium, calcium, magnesium, this kind of. Fifth one is antibiotics. Which kind of antibiotics are used? That is penicillin, kanamycin, amphotericin B, etc. Sixth one is growth factor. Growth factor is glutamine. Seventh one is hormones like penicillin, thyroxine, hydrocortisone, these kind of hormones are used. And the eighth composition is oxygen, carbon dioxide, etc. Their maintained concentration. Okay. Now, what is the environment or what are the factors or requirement? So, factors or requirements. Which type of factors are used? First factor is pH. That should be obviously 7 to 7.4. Second one is temperature. That is obviously plus minus 37 degrees Celsius. Third one is osmolarity. What is osmolarity? Salt, glucose or amino acid which are using, the concentration must be maintained. That is denoting the osmolarity. Okay. The fourth one is gaseous environment. Means the O2, CO2 which are using, we should maintain their concentration. Fifth one is sterile work area. Obviously, you are using cell culture technique, animal cell culture technique, so must be the environment obviously microorganism free, so we need to use sterile work area. Sixth one is incubation facility. CO2 incubators are mainly used. Okay. Seventh one is refrigeration. Or freezer facility. Which type of refrigeration is used? That is obviously cryopreservation. What is cryopreservation? Cryopreservation is when the cells are preserving at low temperature like minus 196 degrees Celsius using liquid nitrogen. This technique is known as cryopreservation. Okay, and the medium which is used in this technique is DMSO or dimethyl sulfoxide. Now, what are the last one is microscope. Now, next part is what are the types? Animal cell culture technique is divided into several categories or several types. First one is primary cell culture. What is primary cell culture? When you are collecting cells or tissues directly from the human skin or the living organism is known as primary cell culture technique. Okay. Second one is transformed cell culture. In this cell culture technique, what is happened? Actually, cells are naturally or genetically manipulated to change their phenotypic appearance, okay, or to change their phenotype permanently. So, phenotype of the cells are changed or transformed. So, that's why this type of cell culture technique is known as transformed cell culture. The third one is secondary cell culture. What happened in this cell culture? It primarily cell culture when the cells are collected from primary cell culture and then subcultured and grow for a longer period of time in a fresh medium that will form secondary cell culture. So what I have said, cells are collected from primary cell culture, then they are subcultured and grows for a longer period of time in a fresh medium that and they thus form secondary cell culture. Okay. So what do you mean by subculture? Subculture is a procedure of transferring cells into fresh medium from the stock medium or stock culture. Okay. Here the stock culture obviously the primary cell culture. Now, what are the tertiary cell culture? when the secondary cell culture further subcultured and stored for a longer period of time, obviously that is leads to secondary tertiary cell culture. And further, the term quaternary, obviously tertiary cell culture, then subculture, and from quaternary cell culture. Okay? Now, what do you mean by cell lines? Cell lines is quite different from this type of cell culture. It is actually applies to a defined population of cells that can be maintained in culture for an extended period of time, which retains the stability of certain phenotype and function. So, cell line, what is cell line? Actually, it defined to certain population of cells that maintained and cultured for a extended period of time with good stability, phenotype, and function. The popular cell line which is mainly used is known as HeLa cell line. It is a cervical cell line culture which is collected from a patient named Henrietta Lacks. Okay. Now, what are the general procedures? First, we will collect the cells from the human skin. So, collecting samples from the living organism. So, collecting cells by moving scalpel. Okay. After collection, mechanical disruption is done by using motor pressing. Second one is mechanical disruption. After mechanical disruption, they are filtered. And the pore size of the filter must be 0.22 micron. Okay. After filtration, enzymatic digestion is done. By using the enzyme like trypsin and collagenase. After enzymatic digestion, the cells are counted. By using Hemocytometer or cell analyzer, anything. And the cells counting must be, to ensure that the cells count must be 1 to 2 into 10 to the minus 5 cells per ml. Okay. After counting, the cells are transferred to a flask of 25 centimeter square area. So, 5 ml of cells transferred 25 centimeter square flux. Okay. Then the flux is incubated. After that, they are observed daily for their natural growth and characteristics. And obviously, media is changed every 2 to 3 days until the cells attain 80% of confidence. Okay. So, thank you for your attention. This is my end of my lecture. If you like my video, please do like, comment, share and subscribe.